For this whole process, start to finish, right now, how many ATP have we built? Not trying to trick you. 2 ATP have been built so far. So far, how many FADH2? FADH2. 1, but remember, cranks through twice. So we have 2 FADH2. How many GTP? 2 GTP. And now, the hard question, and a very important question. How many NADH? Total. 8. I got one answer of 8. 9 is another answer. 7. So we got what? 7, 8, 9. Okay. You can go with 1, sure. It's like Price is Right, right? You'll go with 10? 10? Go. You're like, just go with her. You agree? Brenna agrees. All right, 10? 8? So we got 7, 8, 9, 10. And 1. 12 just for funsies. All right, I'll count now. How many are here? 2. How many are here? How many are here? But it goes through twice. Equals 10 NADH. So far, so good? 10 NADH. All right, remember, FADH2 and NADH carry really excited electrons that want to do some work. Okay? Can I erase this part? Okay, I'll wait for a second.
The only two are the NADH and FADH2.
I'm sorry, could you say that one more time?
The really energetic ones are the NADH and the FGH.
These are the energetic electrons that we're going to use in the next part. The ATP, the GTP, they've all gone off and they're going to start building things on their own. We're getting ready to move these FADHs and these NADHs somewhere else. Good to go? All right. Last step. Step 4, oxidative phosphorylation. Oxidative phosphorylation is where you're going to take high energy electrons from the Krebs cycle and use them to power the electron transport chain. So we've made NADH and FADH2 here in the mitochondrion. Those energy transport molecules are going to go up to the membrane. So I'm going to sort of get rid of this. I'm going to just create what that membrane looks like, okay? So sort of zooming in. This is a really big view of the mitochondrion, the edge of the mitochondria. Here is the lumen on the inside. This is what's called the intermembrane space. So down here is where we have the Krebs cycle. Right there. It's produced a bunch of NADH. It's produced a bunch of FADH2. We are going to move the NADH, carrying its electrons, and FADH2 to the electron transport chain. Now, the electron transport chain is made up of molecules. Generally, they're attached to transmembrane proteins. Oof, another big word. What's a transmembrane protein? That's exactly what, sorry, I know we did learn all of this already. They are proteins that span the membrane. So if you have these things embedded in the membrane, sort of like that, These drop off. They're a highly energetic electron. So if the electron is moving from here to here, what has this molecule been? So it's reduced because it's gaining the electron. So this picks up an electron. All right? That electron can do work. The work that that electron does is it pushes hydrogen across that barrier. Okay? Yes? Against the gradient. That's where you're using the energy. Because what we're going to do is we're going to build a hydrogen gradient over here.
So then would this be an example of like the primary active?
Yes, this is primary active. That's exactly what this is. All right? So you see where it's all coming together, right? Yeah. That's why I had to teach you all that other stuff to get through this. All right. Now, the molecule next to it, this piece going through the membrane, has higher electronegativity than the one before it. So what does it do to that electron? It takes it. So the electron jumps. Then it goes to this molecule. And what does it do? It pushes hydrogen across. The one next to it is even more electronegative than that one. So what does it do? Steals the electron, pushes hydrogen across. Until the molecule reaches, until it reaches the most electronegative element that we have studied in this class, which is what? Oxygen. Oxygen is the final electron acceptor. We've created an electrochemical gradient here. The electron jumps to oxygen, O2, and that oxygen gets converted into water. This is why you breathe. Literally, that reaction right there is why you need oxygen. Yes?
So after you heard the back process in word with Callaway.
I'll go through it again. Tell you what. I'll definitely go through it several more times, truthfully. This big picture. You create an electrochemical gradient, and you've converted oxygen into water. Yes?
What's the pH level?
That's a good question. I don't know what the pH level is in there, because what happens is, almost as soon as you build this gradient, you use the energy of the gradient for the next step.
Okay.
So this is not going to stay high. Yes?
What's Q?
QB, is that listed there?
No, it's just Q.
Where do you see a Q? So that Q is one of these. One of these, but I'm not going to ask you Q and cytochrome C and all of the names of it. I don't. That's too specific for me. I want you guys to understand the big picture, which is you have an electron transport chain powering this pushing of hydrogen across the gradient.
That's all we need something about that electron transport chain powering the membrane gradient.
Right. So big picture, guys. What's going on is you're taking the electrons from the FADH2 and the NADH. Those electrons are getting past these molecules. These molecules are then pushing hydrogen across the barrier. Each time that happens, the electron jumps to a molecule with a higher electronegativity. Until it reaches the absolute final electron acceptor, which is oxygen. I cannot stress enough that you guys understand that oxygen is the final electron acceptor, and that is the reason you breathe. That is the whole reason you take oxygen into your body, is to power this reaction. Say that again.
So what's produced is also accepted or used to produce it?
What do you mean?
I mean, like, it's produced and you also accept it to start the production.
Are you talking about oxygen?
Yes.
So we're not producing oxygen. We never produce oxygen.
It's just
It's become, yeah, it's converted into water, and that's a waste product.
how does it convert into water?
Because when you reduce oxygen, it becomes water.
Like you add an electron and it becomes water?
Because then it becomes more receptive to hydrogen. Yep.
Oh. Doesn't it come even with like O2 like minus for a second and then it
You could look at it that way, yeah.
okay, that just is helpful.
So what you're doing is you're moving hydrogen across this barrier, creating an electrochemical gradient. Where does the hydrogen want to go? You got lots of hydrogen up here, no hydrogen down there. It wants to come back. And it wants to do that because of diffusion, right? Problem is, this is a semi-permeable membrane. It's not letting the hydrogen through. The only way for hydrogen to get through is to go through another molecule, another protein, set of proteins called ATP synthase. So here you might have the ATP synthase popping through, which point the hydrogen molecule stream through, like that. ATP synthase works almost like a water wheel. Do you guys know where a water wheel is in a river? So as you've got a river flowing, if you put a wheel in it, that wheel begins to spin, pushed by the flow of the river. If the river's moving in this direction, the wheel gets pushed that way and up and around. So you're converting the movement of these hydrogen ions into mechanical energy, kinetic, the energy of motion. Yeah.
Secondary acid.
Which is secondary transport. Yeah, exactly right. So you're pushing this wheel around. And as that wheel gets pushed, it's taking an adenosine diphosphate and a phosphate and putting them together. This is extremely efficient. So you're now able to take the energy from these NADHs and these FADH2s, convert it into that, put it into that electron transport chain, push that hydrogen across. As that hydrogen streams through, it converts ADP into ATP. But it's not just a little, it's a lot. This ATP synthase is extremely efficient. It's got three set phases to it. So if you're watching, check this out. So let's say we got an adenosine diphosphate here. This pen is an adenosine diphosphate. Its cap is a phosphate. So the first step, I'll be the ATP synthase because that's how cool I am. The ADP and the phosphate bind to the ATP synthase, phase 1. Phase 2, it starts to spin because of that hydrogen. Then that brings the ADP and the phosphate close together, binding them. As it spins again, that's released, which means you get back to the first conformation where you can bind a new phosphate and a new ADP. So 1, 2, 3. Does that make sense?
Are enzymes affecting this at all?
No, not this step. You don't, and that's the big difference between substrate level and oxidative phosphorylation. Oxidative phosphorylation is where you're using the power of that gradient to power that ATP synthase. Substrate level is where you have an enzyme doing it all.
So ATP synthase is not an enzyme?
I would not consider it well, I mean, it would reduce the energy you need to bind the two. So you could consider it that.
It's an enzyme that has to be powered by something.
Right. Yes?
Is that where recycling comes into the cell?
Recycling as in, like what do you mean by recycling? Release. Yes, you're converting ADP into ATP, and then that ATP, after it is involved in a hydrolysis reaction, becomes ADP again, and it comes back here to get repowered. It's, instead of recycling, think of it more like a battery charger. Yes? This is all step 4, which is the last step. So the net result of the whole, that was it. That was the last step. And by the way, this is the hardest thing you will learn all semester. It doesn't get any worse. Unless you don't like math, at which point maybe genetics is worse. But I think this is probably the worst. Net result is you're now producing 30 to 34 ATP from this process. You're taking the energy of one molecule of glucose, and through this whole step, all 4 steps, converting it into 30 to 34 ATP. It depends on how efficient the system is. there are going to be different environmental factors going on. You may, it may not be hot enough, or maybe too hot. The shape of the enzymes involved in these processes might change, which means it's going to be less efficient. It might. Honestly, I don't know. I'd have to look that up. Yes?
Just out of curiosity, is the 2 ATP, is that also considered?
This is part of that overall, yeah.
Like overall 30,
Yep.
60?
So you're making a bunch, a whole lot.
So most of it is coming from the later steps.
You can see why this is so important. This is what gave us a major evolutionary edge. Whereas all the other organisms were chugging along, just doing glycolysis. We're going to learn that cycle in just a minute. Getting 2 ATP. Once we established this, our ancestors were able to energetically run circles around everything else. That's why all these multicellular organisms you look at, you, me, plants, animals, anything that's got a mitochondrion in it, and churning out this much energy is, in general, really complex and top of the food chain.
Also, like you were saying, some more primitive organisms still don't use this.
Not at all. And we're going to learn about them in just a minute. Now, it's important to note, I was talking about glucose. Bear in mind, this process is not only for glucose. Other molecules come in at different stages. You cannot live on sugar alone, as much as you may want to. Carbohydrates, fats, and proteins are all broken down, but they come in at different stages. So you're going to eventually end up with the same basic parts, but you have to break them down a little to get there. And they might come in after pyruvate, they might come in farther in the Krebs cycle, but they still come into it.
Is there anything that comes before sugar?
Anything comes like
Yeah, so like how you said other things may come in after, does anything
come before it. So what I've gone through is just, this is basically the process of how to break sugar down. So no, nothing comes in this one. But if I was to show you how to break like fat down, it would be, you start off with fat here, and glucose would come in way later on.
Can you get energy from other foods?
Yes, absolutely.
Oh, this one's just the best.
This is just a model. It's for lack of a better word, it's the simplest model.
So you're saying that this aerobic cellular respiration can happen with like another molecule?
Yep, it absolutely can.
Oh, and you're saying glucose is the one that makes the most ATP, but it's probably not the only one you should be using.
Exactly.